In a previous video, we went over the stages of meiosis, but let's do a quick review now. So recall that meiosis starts very similar to mitosis. So the first stage of it is going to be interphase. And interphase is broken down into kind of three subphases. So we have the G1 phase, the S phase, and the G2 phase. And remember that during the G phases, the cell is mostly going through a period of growth, preparing for the next phase of the cell cycle. It's important to note that during the S phase, we have DNA replication. After interphase, the cell goes through the first round of meiosis, which we just call Meiosis I. And the steps of Meiosis I are similar to the steps of mitosis, and we can remember them by remembering the mnemonic of PP mat C. So the first step of Meiosis I is going to be prophase I, followed by prometaphase I, followed by metaphase I. Then we have anaphase I, followed by telophase I, and at the same time we have cytokinesis. Now, before the second round of meiosis, the cell goes through a period of growth known as interkinesis. And it's important to note that during interkinesis, the cell is growing, but the DNA is not being replicated. So DNA replication only happens once during meiosis, and that's during the S phase of interphase. So during interkinesis, we have growth, but we do not have DNA replication. After interkinesis, the cell goes through another round of meiosis, so another round of cell division. And we call this Meiosis II. And once again, Meiosis II has those same steps of PP, mat C. So the first step is going to be prophase II, followed by prometaphase II, then metaphase II, anaphase II, telophase II, and another round of cytokinesis. Now, even though Meiosis I and Meiosis II are both rounds of cell division, different things are happening. So in the previous video, we looked at Meiosis I, and the key thing that happened during these kind of phases of Meiosis I is that the homologous chromosomes separated. During Meiosis II, you're going to learn that during these stages, what happens is that the sister chromatids are going to separate. And if we were to compare meiosis to mitosis, interphase is similar to mitosis. And Meiosis II is similar to mitosis. So now let's take a closer look at the stages of Meiosis II in more detail to see what happens. So as a reminder, interkinesis is this period of time that happens between meiosis I and meiosis II. And it's similar to interphase, but the key main difference is that the S phase does not occur. So we do not have the S phase, which means that the DNA is not replicated again. And this is really important because remember that the end result of meiosis is that we're going to end up with cells that have half of the chromosomes that they started out with. So if we replicated the DNA again, we would end up back with the same number of chromosomes in each cell, and that would cause a problem. So it's really important to note that during interkinesis, we do not have DNA replication, but we do have a lot of growth happening. So the cell is gathering energy reserves. So it's gathering things like nutrients and oxygen to make ATP, because cell division is a really energy consuming process. It's also going to be making proteins. And that's because it needs proteins, it needs enzymes, et cetera, et cetera. It might be gathering other materials in order to make things like new cell membranes and to make the proteins associated with separating their structures, making, lipids and cholesterol to go into the cell membrane, et cetera, et cetera. So we can say gathering other materials needed to make cells. During the stage of prophase 2, the same thing that happens during prophase of mitosis and prophase 1, where the nuclear envelope breaks down so that the chromosomes can be released from the nucleus, a new mitotic spindle forms, and the chromosomes condense so that we can now see them. Prometaphase 2 is also similar to prometaphase in mitosis and meiosis I, in which the nuclear envelope is completely broken down by this point. And in this case, each sister chromatid forms a kinetochore. And remember, the kinetochore is just this little kind of attachment spot right here for the mitotic spindle fibers to attach to. Now, this is a little bit different from prometaphase 1 because remember that during meiosis I, we were separating the homologous chromosomes. And in prometaphase 2, we're separating the sister chromatids. So the kinetochore forms on each of these sister chromatids so that during anaphase, the sister chromatids are going to get separated from each other, and one's going to go to one side of the cell and one's going to go to the other side of the cell. During metaphase 2, the chromosomes, which remember they are these X-shaped chromosomes, which means that we have sister chromatids present. So we have two sister chromatids that are attached to each other. And so these X-shaped chromosomes are lining up along the middle of the cell because during the next phase, which is anaphase, each of the sister chromatids is going to get pulled to a different side of the cell. Now remember that in metaphase 1, this looked a little different because in metaphase 1, we were separating homologous chromosomes. So in metaphase 1, we would have a cell that looked like this, and our homologous chromosomes would be lined up along the middle of the cell. So that during anaphase, when they got separated, one of the homologous chromosomes would go to one side of the cell, the other homologous chromosome would go to the other side of the cell. But note that in metaphase 2, now the chromosomes, we no longer have homologous chromosomes. So our chromosomes are going to line up more like this, so they're not paired up anymore. And then during anaphase, the sister chromatids are going to get separated from each other. So we end up with one sister chromatid going into one of the new daughter cells and the other sister chromatid going into the other new daughter cell. During anaphase 2, the two sister chromatids are separated from each other. So the centromere, which is the protein that is holding them together, is going to break, and then each chromatid is going to be pulled to a different side of the cell. So remember that our sister chromatids, they start out identical, but they end up looking a little bit different from each other because of genetic recombination. And remember that genetic recombination is also called crossing over, and this happens during prophase 1. And then telophase 2 is just where the chromatids, which now we only have one chromatid, so each chromatid is now considered its own chromosome. So these new chromatids, which are now just new chromosomes, are going to reach either side of the cell, and the nuclear envelope is going to reform around those chromosomes. Cytokinesis is also going to be happening, which is going to ultimately pinch the entire cell into two new cells. Now, if you look down here, you'll notice that we essentially end up with 4 daughter cells, because remember that during meiosis 1, one cell gets divided into 2 cells. And then now we're talking about meiosis 2, where those daughter cells from meiosis 1 are all going through their own round of cell division. So we ultimately end up with 4 daughter cells. So the end result of meiosis 2 is that we end up with these 4 daughter cells, and these daughter cells are not identical to each other, and they are no longer identical to the parent cell as well. And remember that during the first round of meiosis, we separated those homologous chromosomes. So now we have haploid cells because we no longer have those homologous chromosomes present.